Hello, and in this chapter 20 is the importance of antimicrobial medications. So in this chapter, we're going to be covering a lot of different drugs and exactly how their molecular mechanisms work to inhibit the growth or kill the particular infectious agent. And so the history here is that before antimicrobials, it was very difficult to treat anybody that might have had a particular bacterial infection. And so before penicillin came around in the 1940s and 50s, the likelihood of death that came with a bacterial infection was really just left up to luck and the individual's immune system and health of the individual. So the very first antimicrobial drugs were documented in 1910, which was a salvarsan, which was a dye that actually was made for paints, but essentially worked also to stop microbes from growing. So it was kind of strange, and they moved it on to protanozil, which was produced by Dow Chemicals, and this was one of the very first treatments for streptococcal infections. However, when they moved it to the in vitro route and actually were trying to do research on it, in vitro meaning in the laboratory, nothing happened. The bacteria were able to grow just fine. So it wasn't until they realized that there were enzymes inside the mice that actually split the protanozil in half, and one half was actually the active ingredient. And so this became one of the first sulfa drugs because the active ingredient was sulfonamide. And so these are still a class of drugs that are used today. Both are considered what's called chemotherapeutic, which typically we think of chemotherapy as being cancer treatment, but in this case, the true definition of chemotherapeutic is any chemical that's used to treat a disease, and so in this case, both are chemicals. However, when we're actually talking about antimicrobials, we just call them that instead of calling them chemotherapeutic. So that's why the name doesn't actually cross very far out of the cancer realm when it comes to lay people. Now, the discovery of antibiotics what involves knowing that a lot of these microbes that are found in nature actually produce their own defense mechanisms. And so the bacterium group Streptomyces, which we don't really cover in this class besides to just talk about it right now, in my 210 microbiology, there's an entire chapter dedicated to Streptomyces. What makes these guys unique is that they are still prokaryotes, but they contain so much information in terms of genes that they almost act like a eukaryote. So they're kind of right there on the threshold of a prokaryote and eukaryote, even though they are actually prokaryotes. With that being said, the Streptomyces are responsible for well over 80% of our drugs that we use for antibiotics. Simply, we as scientists take advantage of the defensive strategies that these bacteria have against other bacteria and use them to our benefit by manipulating them and ultimately producing them in mass at a pharmaceutical company. And so even today, when we're looking for new medicines, we're still resorting back to nature and searching for new microbes that may have a unique defense strategy that we haven't seen before that we can utilize in a pharmaceutical. So when we talk about these antimicrobials, we want to know something. We want to know what their toxicity is. We want that ultimately there be greater harm to the microbes than the host. It doesn't make any sense to give a drug to somebody that is going to be 50% effective at killing the microbes, but 70% effective at destroying the patient's kidney. It just doesn't work that way. So what we want to do is we want to figure out what the therapeutic index is, and we'll talk about how we get to that here in a little bit. But ultimately, the therapeutic index is the lowest dose that we can give to the patient that is actually therapeutic in terms of clearing the bacteria. So why give 200 milligrams if only 100 milligrams is all you need is kind of the idea here. And so this is going to be a lot of work that goes behind the scenes before a drug ever makes it to market. When we talk about the antimicrobials, they fall into two classes, bacteriostatic and bactericidal. Bacteriostatic is the ability to stop the bacterial growth. So a lot of these are going to be drugs that are responsible for blocking peptidoglycan synthesis. And so if you're already a mature bacteria and you already have your peptidoglycan cell wall, these drugs are ineffective for you. However, remember that bacteria replicate every 20 to 30 minutes, and therefore there's a constant barrage of new bacteria being replicated any given time, and so these drugs are good at stopping them from actually making it to maturity. So with the bacteriostatic, we're still relying on the immune system to come in and do its job and actually clear the remaining mature bacteria from the actual patient. Now, bactericidal are going to kill all bacteria. They're not going to just kill the ones that are growing, but they'll also kill the ones that are in lag phase that have essentially made it to maturity. Spectrum activity talks about how effective it is at hitting different types of bacteria. So typically when somebody comes in with an unknown bacterial infection, what we're going to do is we're going to start them on a broad spectrum antibiotic. This is going to be important for anybody that looks like they may be suffering from acute life-threatening diseases where there's absolutely no time to culture for identification. However, these broad spectrum also will disrupt the normal microbiota and destroy them too. So they are a double-edged sword. They're going to be good at clearing, especially if somebody comes in dying, and they're also going to be negative because they're going to take out some of our normal microbiota. So what we ultimately want to do is get the patient on a narrow spectrum antibiotic if possible. These are going to be specific to the pathogen that's actually infecting them, and therefore we're going to have identification from the pathology lab before we can actually administer any of these antibiotics. So typically you start on broad spectrum and then you move over to narrow in most cases, but of course there's always exemptions. Okay, so effects of combination. So here's just 3 different types of ways if we're going to take more than one drug that they can work. So if they interfere with each other, we call that antagonistic. We do not want to give people medication that is antagonistic because it could potentially be fatal or have terrible side effects. If it enhances, where one drug actually helps the other one do its job, we call that synergistic. And if there's absolutely no difference in how you combine them, it's just called additive. So a good additive would be, let's say, taking an antibiotic to clear an infection, but at the same time you take Tylenol for the pain. Those would be additive, where neither one is necessarily going to affect the other. All right, resistance. A lot of bacteria are what we call innately or intrinsically resistant, meaning that they're just born with it. So for instance, the entire penicillin class of drugs is designed to block cell wall synthesis, specifically the peptidoglycan cell wall synthesis, where Mycoplasma does not have a cell wall made of peptidoglycan. Instead, it has a cell wall made of mycolic acid, and because of that, none of the penicillin-based drugs are going to be useful against Mycoplasma. Another thing that makes some sections of bacteria actually resistant is going to be the gram-negatives. So remember, they have LPS, then they have an outer membrane, then they have the periplasm, and then they have an inner membrane, and then they actually have the cytoplasm. That's a lot of levels for a drug to actually make it through. So some of the gram-negatives are going to be resistant to certain antibiotics just because of their structure setup. Now, other bacteria can actually acquire resistance. So when we're talking about antibiotic resistance increasing in bacteria, we're talking about acquired resistance. And the 2 different ways that they can do that is spontaneous mutations. And because they're replicating every 20 to 30 minutes, just imagine how quickly you're going to get down to 20, 30 generations. So we know that evolution is based off of mutations that occur over time. Well, bacteria are replicating so quickly that they're going through generations of ancestors in a matter of a week or so. So any mutations that are acquired could potentially be in a spot that allows it to become resistant to a drug. However, the more common way that bacteria are going to develop acquired resistance is horizontal gene transfer, otherwise affectionately known as bacterial sex. They use a sex pilus, so it's like a pili. It's going to be an appendage that comes off the side of the cell wall in these particular bacteria, and it's going to connect like a bridge to another bacteria. And these bacteria can share their plasmids through this sex pilus, and so we call this process conjugation. And this is also horizontal gene transfer, which essentially is from one person to another, where vertical gene transfer would be mother to child. So the classes that go into antibacterial drugs, there's going to be 5 major classes, and what I want you to really focus on in this section is how we have quite the plethora of drugs available in the beginning, and as we go down further through the list, it becomes fewer and fewer, mainly because of toxicity issues, and we'll talk about those as we go along. So the first class of drugs are ones that inhibit cell wall synthesis, and because most bacteria have peptoglycan, this makes a great drug target because we do not have peptoglycan in our body, at least not part of our host cells. We may have some normal microbiota that are carrying it, but for the most part, this is a drug class that is completely benign to eukaryotic organisms. Therefore, we're going to have an incredibly high therapeutic index for these guys. And so these are the penicillins, cephalosporins, and what we call the beta-lactam drugs. So looking at these 2 pictures down here, you have penicillin, and you see that 4-ring structure that's highlighted there in the middle. That's what we call a beta-lactam in organic chemistry. Now these particular drugs actually go and cleave that site and actually break that ring in half. And if that occurs, then the bacteria is considered to be resistant to beta-lactam drugs. And so we'll talk about that as we go along. So essentially what these particular drugs do that have these beta-lactam is that they competitively inhibit the enzymes that are responsible for putting peptoglycan together. And so what's going to occur, remember, an enzyme has an active site, and the active site is incredibly specific to whatever it is that it's hunting. Well, in this case, the beta-lactams mimic the design of peptoglycan to the point where the enzyme mistakes it, and once it mistakes it, it becomes irreversibly bound to that enzyme and essentially shut that enzyme down. Once that occurs, the bacteria can no longer produce its cell wall, and essentially it'll die. So all of them differ a little bit in activity. The peptoglycan of gram-positives is exposed, so they tend to be much more susceptible to the beta-lactam drugs, where the outer membrane of the gram-negatives block the drug, as we were talking about a couple minutes ago. Some of the bacteria that are considered resistant to ampicillin and penicillin and all the cillins out there is because they actually produce an enzyme called beta-lactamase, which, you guessed it, is an enzyme that breaks down beta-lactam rings. And so if they break that ring, essentially the drug becomes useless. Some other terms that we give the beta-lactamase is penicillinase. However, beta-lactamase is the more preferred terminology for this particular drug. So as long as, or this particular enzyme, as long as the bacteria is able to produce beta-lactamase, then they're going to be resistant to many of the cell wall synthesis drugs. So as you can see here on the side, what we're going to talk about is that the basic structure stays the same. As you can see in the yellow, that beta-lactam ring there and the pentamer ring next to it to the right, all of that stays the same in any of these drugs. All it changes is a side group. So look at the very first one, penicillin G. That was the original penicillin that was discovered in Penicillium cryogenum, which you can see there. That's a natural drug. And as chemists got a hold of it, they were able to start manipulating it a little bit to make it work a little bit better. So penicillin 5 came out shortly after, and with adding that simple oxygen, notice that the only difference between penicillin G and penicillin V is the fact that there's an extra oxygen there. And that oxygen allows it to be acid resistant, so it's going to survive the stomach acid a little bit better to make it to the intestines and be a more effective drug. Methicillin, right there, just simply has methyls in 2 different spots. You have dicloxacillin. I'm sorry, I can't see it. You can see there that there's a couple changes, but essentially as we go through, the entire basic structure still stays the same of all of these. And so each of these little changes actually helps us get around one of the defenses that the bacteria may have that makes it resistant. Both of them have, but all the penicillins have either broad spectrum or extended spectrum. The broad spectrum is going to be useful in actually hitting both gram positives and gram negatives, and it's simply going to be due to the fact that it has had that side chain manipulated a little bit. But again, most of these actually get inactivated by the beta-lactamases that are being brought on board by the bacteria. Extended spectrum is going to be changes that we did to the side chain to allow it to have better activity against a certain species. So in this case, Pseudomonas is a major player in pneumonia in the hospital settings, and here it's a gram negative. Well, we already know that gram negatives tend to resist penicillin, but in this case, this particular drug has pretty good use against Pseudomonas in this setting. Now, penicillins can also be added together with a beta-lactamase inhibitor. So the idea here is a bacteria that may be naturally resistant to a particular penicillin-based drug. Perhaps already is carrying beta-lactamase on a plasmid. In this case, what we're trying to do is we're trying to hit it twice. We're trying to hit it with a penicillin, but we're also trying to hit it with beta-lactamase inhibitor, which is going to do exactly what it sounds like. It's going to inhibit the ability of the bacteria's enzyme beta-lactamase from destroying our drug that we're administering. So again, different strategies here for different infections. It just depends on what we're looking for. Now the cephalosporins get their name because they came from a different fungus, where the penicillins came from one fungus, the cephalosporins come from another. And we've had the same chemical modifications to cephalosporins, yielding about 4 different generations. And these are much more effective than the original cephalosporins. So these are important for certain types of bacterial infections or people that may be allergic to penicillin. One thing I want to point out while we're going through this is that it's not incredibly essential that you memorize every single drug class and every single drug name. What I want you to do is focus on the different classes of drugs we have, maybe some of the more important drugs within that class, and ultimately let me be able to tell me why that particular drug is special. So please don't sit here and try to memorize everything. You might go a little crazy. Some other beta-lactam antibiotics are carbinamins and monobactams. Both of these resist beta-lactamases, so these are good for bacteria that are naturally resistant to whatever drug you're originally giving. And carbinamine has a pretty broad range, while monobactam is used specifically to combat E. coli type diseases. Now, vancomycin, gets a big name because we talk about MRSA, methicillin-resistant Staph aureus, and the fact that if you have MRSA, then you usually have to go on vancomycin. But vancomycin still is just the same. It's just blocking peptidoglycan synthesis, so there's really not a whole lot special to it except for one key fact. The reason why we don't use it as much as we use other antibiotics is because this one is absorbed very, very poorly from the intestines. And so if I was to give you a pill form of this, probably would not get much of it into your bloodstream. It'd be a big waste. However, if I give it to you through IV, it's much more effective. And so in this case, the IV is going to be the limiting factor here. You're not going to be able to send somebody home with an IV. You're going to need to do it in-house. And so that's why vancomycin tends to be kept for the end, is just due to its natural behavior inside a person. So this is an important treatment for gram-positives that are resistant to a lot of the beta-lactam drugs. So this is an opportunity to stand out against it. And also it's going to be very important for Clostridium difficile, which is a intestinal bacteria that causes severe disease in people. So this is drug of last resort, mainly because of what I just went through, and we see that resistance is increasingly becoming a problem, which isn't surprising since it's just another peptidoglycan synthesis drug. Now, bactitracin is something that all of us have in our medicine cabinet, but it's kind of unique because its selective toxicity is so low, meaning that it's incredibly toxic to us. So in this case, bactitracin is found in a lot of our first aid ointments, such as Neosporin and whatnot, simply just as a antibiotic they put in there to help prevent infection in an open wound. But it's limited truly to just topical application, meaning on the skin, because otherwise it's highly toxic to the intestines and to the stomach areas. Now, the next class of drugs that we're going to look at is going to be the protein synthesis inhibitors. And so here we start getting an increase in toxicity, mainly because most of these drugs are going to target the ribosome, which both prokaryote and eukaryotes have ribosomes. However, there is a small subtle difference in the size of the ribosomes where we can get some effective drug treatment. So prokaryotes have what's called a 70S ribosome, and this is just a different way of weighing things. So like, weighing things in grams or pounds or whatever it might be. When we're looking at ribosomes, we're weighing them by Svedberg units, which is just somebody's last name that came up with this system. So something you don't need to really know, but in case you're curious what that means, that's what it's looking at. And us eukaryotes have an 80S ribosome. So there is a little bit of difference there, but not a lot. And also, one of the things we got to remember is that the mitochondria is actually considered to be ancient bacterial remnants, in the fact that it has its own DNA and produces its own ribosomes. And the ribosomes that are found in the mitochondria are 70S, just like prokaryotes. So again, it harkens to the fact that the mitochondria has a bacterial background, and because of that we think that the mitochondria may be a source for a lot of the toxicity in these drugs. So the aminoglycosides irreversibly bind to the lower ribosome subunit, causing it to distort and malfunction. So remember, in case you are not really strong in what a ribosome is, I'll back up for a second. Essentially, the ribosome is where the RNA goes to be translated into a protein. And so the ribosome is responsible for protein synthesis. It has two subunits. It has the large and the small, and essentially the mRNA will go in between. It's almost like a sandwich. The mRNA ends up being the meat between the two pieces of ribosome. And in that time, amino acids are going to be brought to the ribosome via tRNAs, or transfer RNAs, that are going to bring the amino acids one at a time to match up with what the RNA is saying needs to go there. And ultimately it's going to start forming those peptide bonds between amino acids and forming the very beginnings of a protein. And so here the aminoglycosides are going to irreversibly bind to that lower subunit, causing it to distort and malfunction, basically blocking translation, or worst case scenario, or just as bad I should say, is going to be causing misreading of the mRNA by the ribosome. These are going to be pretty ineffective to the anaerobes, and so the anaerobes are anything that grows in areas that have no oxygen, so soil bacterium such as Clostridium species, the E. coli, as well as the Streptococci. Since they enter cells via active transport process, these drugs actually have to be brought into the bacteria, and with these particular groupings they don't bring in this drug very well. So this is also a very toxic drug, so it's not going to be used when there's other alternatives available. But however, if this is like a last case chance, or you tried everything else, then of course we can give you a try and see how it works. One of the other ones that actually has to be, it's so toxic that it actually has to be administered by inhalation, is tobramycin. And this is truly only going to be used for cystic fibrosis patients. And one of the reminders about cystic fibrosis is that the mucociliary escalator doesn't work in these individuals. And so they're going to constantly be battling bacterial infections in their lungs for their entire life. And eventually a lot of these patients just run out of options in terms of what drugs work anymore for what they have infected, because the bacteria in their lungs just continue to become more and more antibiotic resistant. So that's why tobramycin is actually a drug that's only used in cystic fibrosis patients, mainly because of its toxicity, but because of the unique nature of a cystic fibrosis patient's lung setup. It becomes essential sometimes to use these in order to maintain life in these individuals. The tetracyclines are what we call bacteriostatic. They're going to reversibly bind, so this means that they can fall off. This is going to block the tRNA, bringing in the amino acid, thus preventing translation. So again, it really doesn't matter how we block translation as long as translation is blocked. This one here will be actively transported into the prokaryotes, but not animal cells, which makes the toxicity a little bit better for us to tolerate. The glycylcyclines are related to the tetracyclines. Same exact action and effect, however, they have a wider activity and sometimes can actually be effective if the tetracyclines are not effective due to resistance. You can possibly try this other class of drugs and they could potentially work. And however, right now there's only one that's been currently approved by the FDA for administration. The macrolides prevent continuation of translation, so this is going to be one of the first choices for people that are allergic to the beta-lactam drugs. However, we do see resistance to these guys because what they'll do is they'll modify their rRNA target to ensure that the new formed ribosome RNA is not recognized by the drug. Now, chloramphenicol is one that has a lot of research capacity, but not very much clinical. It's a bacteriostatic drug, binds to the upper portion of the ribosome subunit, ultimately blocking translation. It's very broad spectrum, but it is mainly used as a drug of last resort because the potential for a very rare but very lethal side effect where people get aplastic anemia, which means that essentially they have no red or white blood cells because the ones that come out are not able to form correctly. And so this becomes very life-threatening very quickly. These next grouping of drugs is going to just be different variations of what we've already seen. So again, I don't want you getting too wrapped up in all the names as much as just seeing how they work. They're basically binding to the 50S, blocking translation. They're bacteriostatic for some, but not others. So this is why it becomes important for documenting what particular pathogen the person's infected with. All these things can be important for making sure we get the right drug. So now we switched over to nucleic acid synthesis inhibition. And these are going to be ones that are going to block DNA polymerase. Ultimately, every time the cell goes through replication, it needs to replicate its DNA, and these are going to be polymerases that are responsible for that. However, these are going to be drugs that are going to recognize the polymerase or some of the enzymes that are responsible for helping with DNA replication. First one we have here is fluoroquinolone, and so it's one of these that actually inhibits one of the enzymes that's responsible for DNA replication. This is topoisomerase. And so the enzyme topoisomerase is really important for maintaining the prevention of DNA from supercoiling. So remember, DNA is helix based, and so we're having to unwind the DNA in order to expose all the nucleotides for replication or for expression. And so as we unwind it, that pressure is going to want to go somewhere, and so without topoisomerase, the DNA will essentially just supercoil the wrong way and then eventually be trashed, and that's going to be no good to that cell and ultimately to the organism if it happens too many times. And so here, the typical bacteria and eukaryote has topoisomerase in there to prevent the supercoiling from happening and make sure DNA isn't damaged. Now, fluoroquinolones are going to go in and block topoisomerase, thus essentially making it ineffective and now bringing back supercoiling into the picture. So we consider this bactericidal, and it's very good against a wide range of bacteria. It is going to have a little bit of toxicity issue because we have topoisomerases too. So it's not going to be the best first gen drug, but it's going to do what it needs to do. The next one is rifamycins. These are going to block prokaryotic RNA polymerase. So in this case, this is a polymerase that's responsible for producing the mRNA in a prokaryote. If this occurs, it's going to actually block the initiation of transcription, which is from DNA to RNA. And so these are also bactericidal, but the resistance is very quick to develop with rifamycin. As you'll see in chapter 21, when you recover tuberculosis, one of the 3 drugs that they can give to a tuberculosis patient is rifamycin, but it's never going to be given on its own because of the chance that antibiotic resistance is always going to pop up readily with it. Instead, it's going to be used in combination with other drugs. This one here is, the next one is metronidazole, and so this one's really good against anaerobic organisms, so a lot of the soil microorganisms that we've already talked about. It's actually a good drug for them. Now notice there we only had 1 page for nucleic acid synthesis inhibitors because it is such an important role in our body too that we don't want to potentially damage our body by toxicity. So the next one is interfering with metabolic pathways, and again, bacteria are used for a plethora of studies, and one of them is just understanding metabolism in eukaryotes. So a lot of the pathways are going to be identical in humans and also bacteria. So we have to find those very specific pathways that are reported only in bacteria, so we don't find something that's actually going to be too toxic to the patient. And so one of those is folic acid. Folic acid is something we cannot make on our own. We get it from our environment, from our diet, but it's essentially bacteria that are producing it. And this is something that, again, we don't have. And so by blocking the 2 of the enzymes that are important for producing folic acid, we actually will kill off the bacteria. However, resistance to these drugs is pretty high in most bacteria. They're going to have resistance on a plasmid that's going to lower the affinity, meaning how well these drugs attach to their targets. And the genes are going to be found on the same plasmid. So it's not like, if they pick up resistance to one, and they're going to not be resistant to the other. Typically, it's going to cause them to be resistant to both drug strains. Now, another one that's in that same, basically these are the 2 drugs that are responsible for blocking folic acid. One is sulfonamides, which we've seen in the very beginning. They're going to inhibit both gram-positives and gram-negatives because most bacteria are producing folic acid. And then trimethoprim is hitting the further downstream target. And so ultimately, by combining these two drugs, we do have a synergistic effect. And also minimizes any sort of resistance problems that may happen. By hitting a bacteria with multiple drugs usually prevents resistance from popping up logarithmically, meaning it's not just a 1 or 2 increase. It usually ends up being a 100-fold increase in protecting against resistance. So the combination of these 2 drugs are often used to treat urinary tract infections. Now notice that we are now to cell membrane integrity. So again, this is going to be something that we're trying to destroy the cell membrane, the phospholipid bilayer of the bacteria, and therefore we're going to end up having severe problems because our cells have a phospholipid bilayer too. This is going to be something that we're going to have to be very careful with and messing with because, again, we don't want to have toxicity to our own cells. So we have 2 different flavors here. We have daptomycin, which is only going to be useful against the gram-positives. Mainly, it can't get through the 2 barriers of the gram-negative cells along with the LPS. And we're going to use this drug only when all other drugs have been used and found to be resistant, or found the bacteria is resistant to them. So this is a drug of last resort, much like vancomycin. Polymyxin B is going to be used for gram-negatives. It binds to the membrane and destroys them. So those are the 5 major classes of antibiotics. The next class is mainly just for Mycobacterium. And so remember, Mycobacterium are made of mycolic acid instead of peptoglycan. And therefore, any of these peptoglycan drugs are going to be useless. So one of the biggest problems with mycolic acid is it acts like a very waxy substance, more lipid-like than protein at all. And also, because it's very lipid, it's going to block a lot of drugs from actually being able to get in. And on top of that, a lot of these drugs that are bacteriostatic rely on the fact that bacteria replicate so quickly. With Mycobacterium, they are growing incredibly slow. So even the bacterial static drugs really have no serious effect in the short term. So what we do is we usually have to use a combination therapy. This is going to require 2 or more drugs to ultimately decrease the chance of resistance, as I just said in the last slide. And so the 2 drugs that we try to hit are going to be unique for the cell wall of Mycobacterium. One is isoniazid, which inhibits mycolic acid synthesis, and the other one is ethambutol, which is going to also block enzymes that are responsible for cell wall synthesis. So if this is the first time somebody's been diagnosed with tuberculosis or leprosy, these 2 drugs together usually are okay. But what we're seeing in other parts of the world is a lot of resistance popping up to these bacteria, and so we're going to have to resort to second line drugs, which have much higher toxicity problems and usually are actually less effective. So this is where we have problems now with the Mycobacterium, is that as we see resistance increasing, we're not finding effective treatments for secondary drugs. So how do we determine if these drugs are any good? How do we actually get those therapeutic indexes? So one of the ways we do it is what's called one of the things that we do to figure out therapeutic index is what's called a MIC, which is the minimum inhibitory concentration of the drug. So what is the lowest concentration that we can go to prevent growth in vitro? One of the ways we do this is serial dilutions, and if you're not familiar with serial dilutions, there's probably some pretty good YouTube videos out there that explain it, but essentially you're just diluting something. So if I start with 100 and I dilute it by a factor of 2, that means my next one's only going to have 50 in it, my next one's only going to have 25, my next one's going to have 12.5, the next one's 6.25, and so forth and so forth. And so by doing these serial dilutions, we're effectively lowering the amount in the next one by whatever factor we choose. Sometimes it's 10, sometimes it's 2, sometimes it's 5. It just depends on your strategy. So serial dilutions are just one way to dilute out the drug and make a little bit less. And so what we're going to do then is we're going to see if the bacteria are still able to grow in media that contains those different concentrations of drug. And ultimately, once we get to once we get to a concentration that prevents the bacteria from growing, that is going to be the concentration that we determine to be the MIC at that time. So let's say it falls between, in my hypothesis of using 100 milligrams, and then the next one would be 50 milligrams. Let's say it falls at 50 milligrams. The minimum inhibitory concentration is 50. Well, does that mean 60, 70 is not going to work? Or does it mean 40 and 30 is not going to work? Because remember, the next one down from 50 is going to be 25. So we're going to have to go ahead and do further analysis of the ones in between to ensure that we're actually getting the concentration that is the true MIC. Now the next one is the MBC, which is going to be the minimum bactericidal concentration. It is the lowest concentration that kills 99.9% of the cells in vitro. Again, this is going to be in Petri plates. Very similar to the MIC, except for here we're trying to figure out the difference between bactericidal and bacteriostatic. These are very good techniques. I've used them before, but they are very labor intensive and thus expensive, not just from the cost of all the reagents, but the cost of the person's salary doing the techniques. So we've come up with other strategies such as the Kirby-Bauer diffusion test. Anybody that's willing to take the microbiology lab we have here, you will be exposed to this test. But essentially I have a picture of it here in the top. What you see there is a Petri plate. If you've never seen a Petri plate with bacteria on it, that's what that tannish stuff is. It essentially is bacteria growing and has colonized the entire plate, except for those little white discs. Those little white discs are purchased from the companies that produce the drugs, or you can actually buy this paper and ultimately make your own little disc out of a new drug that you might be testing. And so different ways to get it, but essentially all the same is that different concentration of the drug are absorbed on those papers, and you place them on the Petri plate before the bacteria is able to grow. So yes, I played out the bacteria, but it's not going to grow to this tannish color until maybe 12 to 15 hours later. And so any of these discs could prevent the bacteria from growing, and that's what you see around most of them is clearing. And so those are probably all different different concentrations. So let's imagine this one's 100, this one's 50, 25, 12.5, 6.25, 3.125, and then here might be 1.5. So the minimum inhibitory concentration is between these two. What it is actually, we'd probably have to go ahead and figure out, maybe do 5, 4.5, 4, 3.5, whatever, and do another plate. But essentially, that's what we're looking at here. We're looking at different concentrations that are preventing growth, and we want to get to the one that is the least amount of the drug to ultimately stop growth. And so this test is called a Kirby-Bauer disc diffusion test. Another variation of that is what's called an E-test, and so here these strips that are growing on this bacterial plate, or these strips that are placed on the bacterial plate, have a progression of drug concentration on them. So here's going to be the lowest concentration towards the center, and greater concentration all the way up. So then all we have to do is read down, and whatever the line is right here would tell us what the concentration of the drug is at that moment, and that will help us be able to do a whole bunch of these discs all at once. But these are not as readily available. This tends to be the more standard practice, the Kirby-Bauers over the E-test. Now, some commercial modifications have made this much more simpler, where we can basically use machinery or computers and whatnot to analyze a whole bunch of different bacterial colonies all at once. And what used to take a few days to figure out what the minimum inhibitory concentration was now can be done in a few hours if you have the money to purchase one of these larger machines.